For this project, our main objective is to monitor the effects of prescribed burns on mugwort Ceanothus plants by measuring a variety of physiological characteristics. Ceanothus is a shrub from the Ramanaceae family that grows these distinct, dense clusters of blue flowers. They have a strong scent and have the unique ability to be used as a natural kind of soap in the field. When mixed with water and rubbed against your hands, Ceanothus will create a wonderful smelling lather. Ceanothus is also an interesting plant to study due to its ability to form nodules with symbiotic nitrogen-fixing bacteria. Much of the world, and specifically California, is being exposed to severe drought conditions that is putting stress on the native population of plants. Plants that are already equipped with strategies to drought tolerance may have better resistance, but when faced with wildfires or a prescribed burn, it may take a toll on these species. Researching the effects of fires on these plants will help us better understand whether or not they are being negatively affected in regards to stomatal size, plant growth, and CO2 uptake. What we really wanted to know is, what are the physiological responses to fire-stratified plants when exposed to drought-stricken conditions? And what are the differences when compared to a plant that has not undergone exposure to fire but is still being put through the drought? We are using various methods in 2 locations, one near the Upper Campus Natural Reserve that has been subject to a prescribed burn, and an unburnt area near the Arboretum. Our main methods we have chose to look into are the height and abundance, xylem structure, and the water potential. After having selected 5 plants from each plot, we have since been able to collect data on the plant size and abundance thus far. After measuring and counting the plants within a 5x5 plot around, it has been shown that the plants in the upper campus are shorter, but are more abundant, while the plants near the Arboretum are taller, but lack abundance. For the other 2 methods, we unfortunately have not been able to get to that, but we are currently working on it. We are eager to collect stem cross-sections to take to the lab. Under the microscope, we will be able to examine the structure of the xylem and really compare the differences between how efficiently the plants are transporting water. We are also very excited to lug the pressurized pyrometer into the field to get numerical data regarding the plants' water potential. We hypothesize that Ceanothus exposed to fire through wildfires or controlled burns will have smaller size ranges, will be limited in terms of new growth and abundance, demonstrate greater water conservation effectiveness within the plant, and be characterized by a woodier and more sturdy outer layer. These findings would suggest that fire has immense morphological impacts on Ceanothus, ultimately leading to deviation from undisturbed natural morphological makeup of Ceanothus.